Mechanism of absorption of salts in plants.
Plants absorb mineral salts, ions, from the soil through their roots primarily by two mechanisms.
1. Passive absorption.
This occurs without the use of metabolic energy, ATP, and is driven by the natural movement of ions along the concentration gradient, from high to low concentration.
It involves diffusion. Ions move from the soil solution into root cells. Facilitated diffusion. Specific carrier proteins in the plasma membrane help ions cross the membrane. 2. Active absorption.
This process requires energy, ATP, because ions move against the concentration gradient, from low to high concentration.
It involves
ion pumps or transport proteins in the root cell membranes that actively transport ions like K, Ca superscript 2, and NO3 into the root cells.
This allows the plant to accumulate minerals even if they are present in low concentrations in the soil.
Once inside the root cells, the ions are transported to the xylem and then distributed throughout the plant via transpiration pull.
Deficiency symptoms in plants. 1. Potassium K deficiency. Symptoms: yellowing of leaf margins, chlorosis, especially in older leaves. Leaf edges may become brown and scorched, necrosis. Weak stems and poor resistance to drought and disease.
Reduced flowering and fruit development.
2. Calcium Ca deficiency. Symptoms:
necrosis at the tips and margins of young leaves.
Deformed or stunted new growth. Root tips may die, root development is poor. Common issues include blossom end rot in tomatoes and tip burn in leafy vegetables. 3. Iron Fe deficiency. Symptoms: interveinal chlorosis in young leaves, leaves turn yellow, but veins remain green.
Severe deficiency can lead to leaf whitening and poor plant growth.
Common in alkaline soils where iron is less available. Would you like a diagram or table to help visualize these processes and symptoms?
Sterility systems used in hybrid seed production.
Hybrid seed production requires controlled pollination, ensuring that the female parent does not self-pollinate.
Male sterility is used to prevent self-fertilization, and there are several types of sterility systems.
1. Genetic male sterility, GMS. Controlled by nuclear genes.
Male sterile plants produce no functional pollen but have normal female fertility. Requires identification and separation of sterile and fertile plants manually, as male sterility is usually recessive.
Limitation: maintenance of sterile lines is labor-intensive. 2. Cytoplasmic male sterility, CMS.
Controlled by cytoplasmic, mitochondrial, genes, often in combination with nuclear genes. Male sterility is maternally inherited, from the female parent.
3 lines are used.
A line, male
3.
sterile,
Cytoplasmic
used as the
genetic
F.
male sterility, CGMS. Combination of cytoplasmic and nuclear genes. Offers more flexibility and control in hybrid seed production. Fertility restoration is possible by using restorer genes in the pollen parent. 4. Environment-sensitive genic male sterility, EGMS.
Male sterility is influenced by environmental factors like temperature or photoperiod.
2 types. Thermosensitive genic male sterility, TGMS. Photosensitive genic male sterility, PGMS.
Used especially in two-line hybrid rice production systems.
Chemically induced male sterility.
Uses chemical hybridizing agents, CHAs, to induce temporary male sterility.
CHAs are applied to plants at specific stages to inhibit pollen development. Advantages:
no need to develop or maintain sterile lines.
Disadvantages: cost of chemicals, environmental concerns and variability in effectiveness. Potential phytotoxicity to plants if not properly managed. Conclusion:
sterility systems play a vital role in producing hybrid seeds efficiently by eliminating the need for manual emasculation.
CMS and CGMS are the most widely used due to their practicality and scalability, while CHAs offer an alternative but are less commonly used due to management and cost issues.